Okay, so the first group of questions, they're just general questions.
Okay.
Um, so the first one is, what inspired you to pursue a career in microbiology? And what did your path look like to become a microbiologist researcher?
Okay. Um, so what inspired, first part of that question was what inspired me to go into microbiology?
Yes.
Right. Okay. So I took microbiology as a senior in college, and I really enjoyed that class. I felt like I was Sherlock Holmes. Do you know who Sherlock Holmes is? The
He's the
detective.
detective, yes.
Yeah, yeah, yeah. So I had to like find the unknown like microorganism, like kind of understand what it was.
Hmm.
So I felt like a detective trying to identify what this unknown organism was. And my teacher was very engaging, and so I really enjoyed my microbiology class. So it was a lab and a lecture.
That's awesome. I'm currently
Yeah,
taking that class and I love it too.
nice. Yeah, yeah, yeah. And so I was like, oh, I want to learn more because my microbiology class was more clinical based, and so I
Hmm.
want to know more about the microbes. And so I was like, oh,
Yeah.
let me go into this field. Um, so a lot of the graduate programs I applied to were microbiology based,
Hmm.
even though I was a chemistry major and a bio minor in college.
Awesome.
Mhm.
And then what led you to teaching since
Oh,
you're a research researcher?
yeah. So the teaching portion comes from I've always been, I guess, a natural teacher. I've always like kind of helped people out. When I was in college, I did tutor some of my peers in chemistry, and then I formally tutored later on when I was a graduate student. Um, I love education. I kind of just love teaching people.
That's
Um,
awesome.
I had a professor who was teaching, I believe it was either inorganic chemistry or physical chemistry, and he just didn't teach us at all. I didn't understand anything. Google actually taught me that class. Um, and so I was like destined in like my heart. I was like, I'm gonna be the best teacher I could ever be. I'll make sure my students understand all the material. And so I definitely wanted to teach, and to be a professor, you need to get a PhD.
Yeah.
So you had a second part of the question.
Oh yeah. It was, what did your path look like to become like the microbiology research assistant?
Right. Okay. So I had research experience when I was a undergrad. So I did my freshman year, I joined the lab, but I was like a tech. I kind of like washed dishes,
Oh
fill
yeah.
pipette tips, the grunt work. And then the second
Hmm.
year I was in that lab, they gave me a little bit more. I can like plate yeast cells and things like that. And then when I was a junior and senior, I was in a prostate cancer lab. So I was looking
Wow,
at
that's
like
cool.
mouse cells. But I don't like working with mice. They like you gotta kill them.
Oh.
So I was like, no, thank you.
Hmm.
Um, and when I was a senior, I applied to graduate school and I didn't get in, and that was heartbreaking. But my friend, he was telling me about doing a post-bac program. And so the NIH has a post-bac program in Bethesda, Maryland, but also they give money to schools around the US to host post-bac programs. And so I applied to the ones, I think at Baylor, University of Missouri, U Chicago. I don't know if I applied anywhere else. But the one at University of Missouri and University of Chicago accepted me, but I ultimately chose to go to University of Missouri, get out of my comfort zone, no longer
Yeah.
in the Chicago area. Um, and when I was there, they did allow me to do rotations. So I did two rotations. Again, one with the mouse lab to make sure I didn't want to work with mice, and
Hmm.
then one with the strictly more bacteria lab. And so I chose the bacteria lab.
Hmm.
Um, and so I did the post back for a year. And in that one year, I did apply to graduate school. I applied all over the Midwest, basically. And
Hmm.
I got into Indiana University.
Oh, nice.
And so at Indiana University, that's where I did my graduate work. I did that
Hmm.
for 6 years. The first 3 years, I was in a lab. I didn't feel supported in the way that I thought I should be. And so I switched labs, which is somewhat taboo. If you ask people like, "Oh, you shouldn't switch labs," blah blah blah. I
Hmm.
think it's okay to switch labs. Um, Sometimes it's necessary. So I graduated in 2019, and then I did what's called a postdoc. Have you heard of this before? A
The
postdoc
postdoc.
position?
I think so.
Okay,
Okay.
it's like additional training that you get under a mentor after you get your PhD.
Oh, okay.
And so I started my postdoc in August of 2019 at the NIH with Dr. Gigi Stores. And there I started working on the small protein research, which my lab works on now. Um, and also when I was with Gigi, she knew that I was really interested in teaching. And so I did have the opportunity to teach as an adjunct professor at the local community college, which was really fun. So I
Yeah,
taught
that's
both
cool.
a lab and a lecture for intro biology.
Nice.
Um, and I also applied to positions. Ultimately, I did get recruited to Cornell, and so I applied to their program. And then that's why you'll see I'm an assistant research professor because right now I'm 100% research
Mhm.
versus needing to teach. And so when I start I'll start my tenure track position in January 2026. I will be given a assignment to teach to the undergrads, I believe, or grad students.
That's awesome.
Yeah, wherever they need me. So,
Yeah, that's cool. So the next question, you might have answered some of these, but it was how did you navigate the challenges of building a career in microbiology, especially when it came to gaining the relevant experience and finding job opportunities that fit those requirements?
okay. I think it's all about networking.
Hmm.
Um, because even for my postdoc, I didn't apply for postdoc. I went to a conference and Gigi saw my talk and invited me to come out to the NIH to give a talk. And anytime anyone asks you to give a talk, just assume it's a job interview. So, because I didn't know I was getting interviewed for a postdoc position.
Oh, wow.
Um, yeah, so definitely networking, building your network. Can you read me the question one more time? I just want to make sure I hit every point.
So the first portion was just asking how you navigated the challenges that you faced.
Mm, okay. Having a sense of community. So I have my friend Melissa Lamana. Um, I call her my sister from another mister. So when I was in grad school, me and her, we hung out all the time, right? We talked about class. Was it good? Was it bad? Different professors. So, you definitely need some type of friend group. And so, it's me, Mel, Bryn, Britta, Courtney, and Kate. And so we're kind of like this trifecta of ladies who got our PhDs and we
That's
were
so
all going
cool.
through grad school together, right?
Mhm.
Um, so you need that type of support. Outside of family support is great as well. But sometimes they don't always understand what you're going through because they're not in the PhD program with you. And so you need people in the trenches with you so that you can feel supported. Um,
Mhm.
and when I did my post back, there was only three of us who came in at the same time. So we kind of just hung out together and made our own little community. We would have like Friday dinners together and stuff. So that was
Aw.
really fun.
Yeah,
Yeah.
that's awesome. Okay, so then I'll move on to the next question.
Okay.
Um, what are the most important skills for someone starting a career in microbiology research to develop? And then the second portion is how did you acquire these skills?
Um, I think the number one skill is being curious.
Mhm.
Um, I don't know how you would build curiosity per se.
I know it's kind of
But
natural.
yeah, so um a lot of what I do or a lot of what I have done is because I was curious about the subject.
Mhm.
Or I found some type of subject interesting. Um, I think people can learn how to pipette. You don't need pipetting skills. You can go to a lab and learn that. You can learn how to pour plates.
Mhm.
Um, you can learn how to streak out bacteria. A lot of the molecular biology that I do in my lab can be taught. So you don't have to know these skills before you come in. So I do have an undergrad. She's about to join my lab. She has no skills, and that's fine
Oh,
because
okay.
I plan to teach her these skills when she joins the lab. Um, so I'd say for a basic microbiology lab, you don't need many skills before you join because you should be able to be taught these skills once you join. Um, if you, let's say you were a graduate student, I wouldn't expect you to have tons of skills. Maybe just basic pipetting. Maybe
Mhm.
you know how to pour a plate, you know how to streak out bacteria. Um, but outside of that, I don't expect much. But if you are a postdoctoral fellow, okay, I expect you to know how to run experiments, um, have minimal oversight. So it's kind of like at each stage, I would expect different things.
Mm-hmm.
Um, but just generally, if you want to get into microbiology, I just say be curious.
Well, that's awesome
Yeah,
and helpful to know
yeah,
because I feel like a lot of
yeah.
like undergraduates are scared to like
Mm,
join things because they're like, oh, I don't know as much. What if they don't
right,
want me? So that's
right.
cool.
Yeah. No, you shouldn't. You shouldn't feel. I guess it's kind of like imposter syndrome. Like, don't
Hmm.
feel,
Yeah.
um, yeah, that you can't learn. But also, a lot of people know that since you're an undergraduate, we already know you don't have the skill set or the knowledge that a
Hmm.
graduate student would, and that's okay.
Yeah.
Yeah.
Okay. So the next question is, can you describe a particularly rewarding project or research study you have worked on in the field of microbiology?
I think it was my grad work. I was really satisfied with how that project went. And I'll tell you a short story.
Hmm.
So when you're doing research, you need to look into the literature to see, okay, what else has been done? Is there any similarities? And so in doing so, I found out about this protein that could regulate what's called bacteriophages. So these are bacterial viruses.
Hmm.
And I had told my boss at the time, I think this regulates what I'm working on. And he was like, no, don't even go down this avenue. And I didn't listen. I was like, I'm gonna see, I'm just test.
Yeah.
Um, and comes to find out it was like a key player in the regulation of the protein I was studying. And so I felt super good about listening to myself,
Hmm.
and listening to my gut. Um, Sometimes you need to have defiance in science because PIs don't know everything. Um,
Yeah.
but I was also given the freedom to do such exploration, right?
Hmm.
Um, he didn't like stifle my creativity. And so that was really nice. So I felt really good about that. I was
Yeah,
like,
that's really
yes,
cool
look at
and
me.
exciting.
I know some things.
Yeah. That's awesome.
Yeah.
Okay, so the next set of questions are going to be
Hmm.
science-based.
Okay.
So the first one is, how does a strong background in biology contribute to your work as a microbiologist? Or yours was basically because you chem majored, right?
Right. So it didn't help at
Yeah.
all. It didn't help. It really didn't help. Um, they struck.
Yeah.
I actually, when
We
I
could
was at
change.
Indiana, no, 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 it's fine. When I was at Indiana University, because I didn't have like a formal microbiology like bachelor's degree, I
Mhm.
actually took microbiology with the undergrads.
Oh,
I took
wow.
the microbiology course, yeah, to get the
Like the
the
fundamental.
baseline right that I needed. Um, so in my post-bac, I did take molecular biology. And so
Hmm.
I knew those concepts, and I was also doing molecular biology techniques in my lab while I was a post-bac. So I knew
Okay.
those types of skills. But just fundamental microbiology, even though I took the clinical portion, it didn't get into like this is how bacteria grow, and this is how they have these DNA enzymes and these
Hmm.
proteins, and like the clinical microbiology was more of like this organism causes this disease.
Okay.
So that's how I was lacking in that. And you could take classes to help as a graduate student.
Oh, well, that's cool. I didn't know that.
Mhm, 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 mhm.
Let me see, because these are basically biology
No,
questions.
that's
The
fine.
next
You
set
can
is,
ask them.
I
Yeah,
can
ask
ask
them.
them?
Yeah, yeah.
Okay. What role does genetics play in your daily work as a microbiologist, and how important is it to understand the genetic makeup and of biology of microorganisms?
Um, so genetics definitely plays a role.
Mm,
Usually if we're making mutations, we want to make sure we're making the right mutations. We also want to make sure that the strain or the bacterium that we're working with don't have mutations before we start working with them.
mm,
And so I feel like I do genetics almost. every day. Um, I'm not looking at like how they inherit genes, but I'm more looking at how if I make a mutation, how does that change the downstream outcome?
Oh, okay. So is it like, do you get to look at the code sequence or is it
Yeah, so
just,
I
yeah.
do I do look at the DNA code.
Oh, okay, that's cool.
Yeah, yeah, yeah.
Okay.
Um,
Is there anything else you need to add or is that?
I was gonna add like bacteria, they when they divide, they make two identical copies of themselves.
Oh, wow.
And so you don't like inherit any genetic variation um a lot of times unless it's coming in from the environment.
Mm,
hmm
okay. And then the next question is how has modern technology such as advancements in microscopy and DNA sequencing impacted the field of microbiology?
It's definitely opened it up. I think the advances in microscopy are like amazing.
Mm-hmm.
Um, we can freeze samples of bacteria and look at their structure. Um, we can look at how they grow and divide. There's like a time, it's called time lapse. So over time, you can make movies of bacteria in these channels
Oh,
and just
wow.
see how, like, if you give them different media or give them different like antibiotics, how do they respond? And so that's really cool. That's microscopy. And
Mm-hmm.
then you said, what was the other one? Um,
Um, DNA sequencing.
DNA sequencing. Yeah, there's lots of DNA sequencing. If you look on NCBI, which is the National Center for Biotechnology, NCB, let me see. It's with the National Library of Medicine with the NIH. I don't want to get it wrong. National Center for Biotechnology Information. So if you go to
Oh, yeah.
NCBI,
I
yeah.
love that
Um,
website.
Yeah, so if you go, there's like all these genomes of all these organisms, right? And not even just bacteria, but all organisms.
Mm-hmm.
Um, but with bacteria, they keep sequencing and they keep sequencing and they keep sequencing. So we're getting to know more about the organisms that are around us.
Mm-hmm.
Um, but still a lot is unknown.
Yeah, bacteria is a lot. There's a lot.
Mm-hmm. Mm-hmm.
Okay, so the fourth question is, can you explain the importance of microbiology in the development of antibiotics and how your work
Mm-hmm.
contributes to this area of science?
Yeah. Thank God we have bacteria that fight other bacteria.
Mm-hmm.
So if you look at most of the antibiotics that we have, they're not synthetic. They actually come from other organisms that they naturally produce these chemicals to fight off other organisms. So Streptomyces venezuelae is one of many of the organisms. Streptomyces. I'm gonna drop the name in the chat for you.
Oh, okay, thank you.
Um,
I think I've
this
heard
is
about
one
it.
of Yeah, it's a dwell or soil dwelling gram positive bacterium. And it makes antibiotics to kill off other bacteria. Let me see. So this is one. Most of the antibiotics we get come from this Streptomyces class.
Mhm.
And they're actually really cool to look at. Like they make different colors based on the compounds that they make.
Oh, that's
So
awesome.
I'll give you this one as well. Um, and so I don't want to say it was like happenstance, but like some researchers, if they're looking for new antibiotics, they'll challenge two different organisms and they'll see like, oh, this organism doesn't grow very well. This other one may be making something to inhibit the growth.
Hmm.
And so that's a lot of times of how we get antibiotics. Um, if you're working with a chemist, they could potentially produce chemical compounds that you could test to see if they are or if they have antibiotic properties.
Hmm.
Um, But if nature is giving it to you for free, I'd take it,
Mm-hmm.
you know.
Cool.
Hmm.
Okay.
So
And
streptomyces, they have said, produce over two-thirds of the clinical antibiotics that we use.
oh wow, that's pretty big
Yeah,
portion.
yeah. Um, and my research, I don't necessarily look at how small proteins could be antibiotic or if they have antibiotic properties,
Mm-hmm.
but someone could potentially do that in the future.
Oh wow, that's awesome.
Mhm.
Okay. So the next question is, how does an understanding of biochemistry help you in studying the metabolic processes of microorganisms?
Um, you need to know the fundamental enzymes. And
Mm-hmm.
so when I was a grad, I always go back to grad school. I took biochemistry as an undergrad, and I learned about proteins and amino acids and that type of stuff. But I did take metabolism, a biochemistry metabolism course for microorganisms in grad school. And I felt like that was very helpful because it helps you think about how organisms are taking up different energy sources and then using that in their systems. I really didn't need it when I was in grad school, but I
Mm-hmm.
use it a lot now because I'm looking at how nitrogen flux occurs within an organism. And
Mm-hmm.
so if I didn't have that basis from grad school, I may be like, oh, I don't know how to look for things, or I don't know what this means, but um, just having that kind of basis or that basic knowledge, you can then build upon it.
Yeah, that's good.
Mhm.
And then the second question, what role do chemical reactions play in the study of microbial stress response, and how do you analyze these reactions?
So, so you want to know the role of chemical reactions in bacterial stress responses?
Yes.
Hmm. There's definitely some reactions that occur.
Yeah.
Let's see.
Or are there any specific scenarios where like there's stress related in the bacteria?
Right. I mean, there's all different types of stressors.
Mhm.
I'm just trying to think of
I know there's a lot of
a
variables.
straightforward example. Um, so let's say we're growing our bacteria and they are in a condition where there's low nitrogen, so like low ammonia.
Hmm.
So the cell no longer is able to take up ammonia from the environment. That causes a change internally for the cells, and that change is recognized by the flux of the TCA cycle.
Hmm.
Have you heard of TCA cycle before?
I don't think it's like
That's okay.
yeah.
This is the way that carbon is recycled within bacteria. It's called
Mhm.
TCA cycle. Um, and so a protein is able to pick up on that change and leads to a chemical change. So you need to know biochemistry
Mhm.
of this protein. And so the protein then gets what's called phosphorylated. That changes the structure. And then that leads to additional changes downstream. Um, that phosphate is then passed on to um, another protein, which can then activate certain genes. So you definitely need to know about like protein structure.
Mhm.
There's different phosphorylation sites. Um, so that's just one example of how a stressor can change um, the organism. But you can have oxygen stress, you can have acid stress,
And then there's
UV
nitrogen,
stress.
right?
Right. Yeah,
Yeah.
yeah, yeah. There's a lot of them.
Hmm.
Um, so this, I'm gonna send you the link. This is the TCA cycle.
Okay. I
Um,
want to say we're probably like getting towards that part in our microbiology class.
nice. Okay. Yeah.
Okay, so the next question is, can you explain how the chemical properties of cell membranes affect the interaction? of antibiotics with bacterial cells?
Mm.
I know that one goes back.
So, you know, there's two different types of membranes for bacteria.
Hmm.
So there's gram negative and there's gram positive.
Yeah.
So gram negative, they have extra decoration and they
Mhm.
don't have their peptidoglycan isn't just exposed. There's another membrane with the decoration. Um, that decoration, um, I believe, produces a negative charge, which then can help repel antibiotics. So a lot of gram-negative organisms can be antibiotic resistant because of this.
Mhm.
Or let's say you give them an antibiotic, they recognize this, and then they'll change the outside of their membrane to combat the antibiotic. Um, Gram-positive organisms, they have a thicker peptidoglycan layer, so it does take more um, to like penetrate through.
Mhm.
Um, but they don't have that, I guess, defense system of like changing their um, proteins and lipids on the outside to fight off the antibiotic that's present.
Mhm. So it just like sinks through. Does this one
Hmm.
have to do with like the saturation, like desaturated?
Yeah, that all
Yeah.
plays a
A role.
part. Yep.
Okay, so the last question is, how do you
Hmm.
apply concepts of organic chemistry when studying the synthesis of microbial products?
Hmm. I don't. I don't use any organic chemistry.
No, you don't.
Um, I don't use any. Yeah, because I'm not looking at how they're synthesized. I think
Do you use inorganic or no?
mostly all the tools I use are like molecular biology. Um, Let me see, molecular bacteriology. Let me see.
Okay.
Let me see. Um, yeah, I basically use a lot of molecular biology, and I'm gonna send you a link about
Okay.
this. There are times where I would need to interact with chemists and other people who are in these fields.
Mm-hmm.
Um, and so definitely they're useful. I worked with a person in my postdoc, and he was a PhD. He got his PhD in chemistry. And so I just think he's the smartest person I know. But just knowing like basic properties, like make sure you don't mix these acids together, right? Or
Yeah.
um, these proteins behave well in this type of buffer. Those are types of chemistry things that we would need. Um, Organic chemistry, yeah, I'm not synthesizing anything per se. I'm letting the organisms do that.
Yeah.
Um, but there's definitely you can get a PhD in pharmacology and you're synthesizing drugs and you need to know these types of reactions and stuff like that.
Mm-hmm.
But the work that I do doesn't require that.
Okay, that's good to
Yeah.
know.
Yeah.
And then you're at Cornell, right?
I am.
That's awesome.
Yeah, thank you.
Mhm.
Be 1 year in January 1st.
That's exciting.
Yeah.
Okay. Well, that concludes all the questions that I have for this interview.
Okay, perfect. And if you need any clarification, please feel free to reach out. And I'll try to reply before Monday.
Oh, yeah, of course. Thank you for
Yeah.
meeting with me again.
Yeah, you're welcome.